I'd like to welcome Kim Morris. Kim is part of our Parents 2.0 cohort. She lives in Kansas City with her family, which includes her adorable 26-year-old daughter, Allison, who has CTD. Since Allison's diagnosis 15 years ago, the Morris family has learned from her and worked tirelessly to advocate on her behalf. They remain hopeful for a better future for everyone affected by CCDS, thanks to this community and its dedicated researchers. It's my pleasure to welcome Kim to share her story with us.
Hello, I'm Kimberly, and I get to tell you about our spunky, lovable, unpredictable Allison. She is 26, and we live in the middle of the United States where it's big-time World Cup soccer activity right now. She received her CTD diagnosis 15 years ago at the age of 10. Her genetic report showed some complicated information, and her MRI showed limited peak. Her CTD is de novo, and she has global symptoms of the disease. We were told she was one of the first girls diagnosed with CTD in the world. Back then, there wasn't very much information about CTD, and there didn't seem to be the blossoming scientific interest as there is right now. And for that, we're so grateful to see it emerging. I've worried about Allison since day one. She's our firstborn, so that's what new parents do, right? These days, my worries include her increasing mobility challenges, our two-story home, and that she's one fall away from big-time issues. I'm concerned, as she just turned 26, that her health insurance coverage has changed and how that impacts the providers she gets to see and the financial impact involved. I weigh out looking into residential services that Allison now qualifies for and worry that that will upset her as well as other family. I worry that we're going to stand out for all the wrong reasons when we're out in public instead of fitting in, which I prefer. The list is lengthy. Every ACD or extraordinary symposium I attend, as well as the updates we see, are so encouraging and emotional. This is hard work, and together we can really do hard things, so thank you for being part of this community. We're hopeful that meaningful change through treatment means Allie can tell us when and where she hurts. Yes, when she falls and when she's ill, but also as well as monthly female processes. It would be amazing if her communication became more articulate and others could understand her as she's trying so hard to express herself. For example, that she's really saying no thank you and not I don't like you. Another meaningful difference for Allison could be improved emotional dysregulation. Her behaviors flare more often, unexpectedly. She's usually able to be redirected and taken from her hot state to a cooler one, but it's definitely a daily struggle and a yo-yo that we just hang on for multiple triggers. On really rough days, she's not just vocally upset, it escalates to hitting, throwing, and biting herself. So any support to reduce those behaviors could be life-changing for her and those around her. We envision the possibility of Allison being able to take a shower or bathe alone, tend to her self-care independently, or with supervision instead of the current 100% support. Our Allison has a smile that lights up the room, a laugh that is contagious, and amazing manners. She says a lot of please and no thank yous. Allison gives great hugs, sometimes to unsuspecting recipients. Who here in this room has been a recipient of one of our girls' hugs? Check it out, you guys, that's special. She has strong skills with puzzles. Allison feels the Holy Spirit at church, and she loves that worship band. She gets really excited for sleeping in a hotel, going to Camp Barnabas, and an Imagine Dragons concert. Our family is very much here for you, and we're cheering you on and hoping to support you as you figure out this strange and complex diagnosis. Thank you.
my pleasure to introduce Thomas Joudinaud and have him come up to the podium for his talk. Thomas is the co-founder and CEO of Ceres Brain Therapeutics, a cardiac surgeon by training. He led strategic consulting experience at Boston Consulting Group, AEC Partners, before he started this company. He holds the distinction for having initiated the first ever clinical trial specifically for CTD. And it's our ultimate pleasure to have him here to speak to us about an update and next steps. We cannot be more grateful for you to be working on this and sharing this update with us. Thank you so much, Thomas. Over to you.
Thank you, Sangeeta. So hello, everybody. I'm very happy to be here and happy to share the result of our work. I talked to you 2 years ago. I talked in front of you 2 years ago. And so I will present you what we have achieved in these last years. So just to remind everybody, and for those who don't know Ceres or don't know me, I'm a medical doctor by background. We created Ceres in 2019. We are a spin-off from an academic center in France. And we are developing CBT-101. It's an innovative creatine prodrug. You have heard about prodrug yesterday a lot. And so this prodrug is entitled to pass the blood-brain barrier. As you know, the systemic creatine, the creatine in the blood, is not going to the brain because the transporter is not expressed at blood-brain barrier level. So whatever you can read in the press, whatever you can understand from the creatine companies, the creatine oral is not going to your brain. So what we are doing is we have a nasal spray, and from this nasal spray, our creatine prodrug is entering the olfactory and trigeminal nerves, which are a direct access to the brain. From the olfactory and trigeminal nerves, it migrates all on those towards their origin, the olfactory bulb and the trigeminal core, and from there it diffuses everywhere in the brain, where it releases creatine in brain neurons. And within the neurons, it can exert their energy activities, as it was explained by most of the speakers today and yesterday. So our idea is that we deliver a small quantity of CBT-101, and thus a small quantity of creatine every day in the brain, specifically in the neurons, and this idea is to supplement the brain every day. It will give enough energy to the brain, according to what we think, for the brain to function normally. So this is all the key achieved milestones since the inception of the company. So as you can see, we started in 2019. We did some experiment on creatine-deficient mice from Matthew Skelton, but also from the one from Laura Baroncelli. We also worked with non-human primates or monkeys. We get an orphan drug designation by the EMA and the FDA. We worked a lot on the formulation. The CMC is a nightmare for small and large companies, and we made more than 2-3 years to get a final formulation that was adequate for human use. Starting in 2023, we did some regulatory toxicology study to be sure that we will not be toxic for the brain and for the patient. 2024, we started also to produce our GMP batch, good manufacturing batch, meaning that what we produce is exactly what will be in the patient, and each dose the previous day will be the same as the day where you take the drug, and the same as the following days, the following days. We get also a pediatric designation in CTD from the FDA. And in 2025, we started our first clinical trial. It was a phase 1 in healthy volunteer, and I will show you our results, more or less. So just to remind everybody also, in monkey, we demonstrated that our drug, CBT-101, reaches the neurons and delivers creatine in the neurons. And after 30 days of intranasal treatment in the monkeys, we were able to show that 50% of the creatine in neurons was coming from our drug. We know that because we used a drug that was labeled with stable isotope. We worked on CTD models, and we demonstrated that we improve cognition using the novel object recognition test. We improve also the brain metabolism of the mice, and we improve the synaptic plasticity using some synaptic biomarkers. We also demonstrated using some other model of neurological disease that we are neuroprotective in neurodegenerative disease. So we had a consistent efficacy across metabolic, genetic, and toxic model with CBT-101. So our phase 1 study, I talked about it last year and 2 years ago. It was made on 48 volunteers. It was a randomized, double-blind, placebo-controlled study. We had 3 cohorts of patients in the single ascending dose, 3 cohorts of patients in the multiple ascending dose that lasts for 14 days. In each cohort, 6 patients received CBT-101 and 2 patients healthy volunteers received placebo. The primary objective of this phase 1 was safety and tolerability of the nasal spray. And we had an exploratory readout, which was quantitative EEG. The idea was to assess if this extra boost of creatine in the neuron could be seen in the healthy volunteer. So on the right-hand side of the slide, you have the protocol here, sorry. So it was, single ascending dose as well as multiple ascending dose. We started with 1 puff every in each nostril. Second cohort, it was 2 puff morning and night in each nostril, and the third cohort was 1 puff in each nostril, more in the morning, at lunch, and at night. We did not have any major adverse event. Some healthy volunteers reported transient nasal tingling or rhinorrhea. And since our drug is really going from the nose to the brain and not in the blood, or when it goes to the blood, it just hydrolyzes itself and disappears in a matter of seconds, we didn't have possibility of quantify the drug in the blood, which was completely consistent with what we observed in the preclinical studies. And we also analyzed the quantitative EEG signal. So this is the methodology that we used. We used an electroencephalogram with a standard electrode positioning, and we recorded the signal both in eyes closed and eyes open condition at day 1, day 2, and day 13. We measured a ratio between the pre-dose recording and the mini EEG activity under CBT-101 and the mini EEG activity under placebo. So it allows us to generate brain maps of this ratio, and then we created a color coding of these brain maps showing the statistical significance of these brain maps or not. Unfortunately, I cannot show you the result today because we are patenting some results, and if I show you, I made public this result, and it creates a notoriety. So I will just explain to you what we have seen. We have a significant difference between the healthy volunteers that received CBT-101 or the placebo in each areas, each electrodes, and at each dose, meaning that already after day 1, with 1 puff in each nostril, we have a change in the beta band power, as well later at day 2 and day 13 in the gamma band power recording in each electrode. And this means that this extra boost of energy, of creatine energy, is seen for the brain, or is made for the brain as an increase in the reservoir, and that the brain has a state of better attention control, he has a cortical activation, which is in a better way. And for us, it signs the fact that our drug is really going to the brain and really going to the neurons, which is, after all the mice, all the dogs, all the monkeys, the first time that we demonstrate that, yes, our CBT-101 is really going to neuron, and yes, there is an activity of this extra creatine. So based on that, we have strong confidence, and it will do something in patient, hopefully. So where are we standing now? We have achieved the CMC. We have a robust manufacturing process. We need to transfer the API process to a new facility which will be FDA approved, and this is ongoing. It will be finished at the end of the year. All the tox study has been done in dogs and rats, and we have achieved the phase 1 study. Since we will treat children, we need to run a toxicology in juvenile animals. So it is ongoing. We have a study that is already conducting in India, actually, because it's much cheaper in India, and it's on juvenile rats. It has been going for 3 months, and we don't have any problem in these rats. And we plan to launch a phase 2/3 study in CTD in 2027. We need to do that to have the results of the 6 months tox animal study, and we also need to have transferred the API batch. Between now and the phase 2/3 study, we will ask to the EMA for scientific advice to validate the protocol of the study. So this is the CMC upgrade. As I said, we are transferring the manufacturing process. It's essential because it needs to be almost a commercial product or the commercial products that we will use during this phase 2 study. And we do that because we would like that this study will be pivotal, meaning that if it's positive, the drug can be approved by EMA and hopefully by FDA as well, because we'll do also an IND meeting with FDA explaining what we are doing. In ultra-rare disease, they may agree that a European study can be, can serve for the US market. This is the ongoing 6-month juvenile toxicology study in rats. So it's a large study, 5 groups of animals. We're talking about more than 200 rats. A group of control, a group of placebo, low dose, medium dose, high dose. So far, no problem after 3 months. Our phase 2, we will leverage the single case experimental design methodology to conduct it. So the idea is that each patient will be his own control. 24 patients should be enrolled, and there will be 6 months treatment. Since we want to treat children, and hopefully the EMA will agree, we will do for the smallest children 2 sprays per day. For the children between 10 and 30 kilograms, 4 sprays per day, and 6 sprays per day for the children that are weighing more than 30 kilograms. This is preliminary. It may change according to our discussion with the experts as well as with the EMA. The idea of a single case experimental design is that we will have a first part of the study where we will observe the patient under placebo. And we'll do a test that we will repeat. And then we will randomly and double-blinded introduce CBT-101. So the patient will receive the placebo in the nose. Then after a few months, and I will show you what we have in mind, he will receive CBT-101. And then we will repeat the test every 2 weeks, 3 weeks. It's under definition with Aurora in particular. Regarding inclusion, we are talking about including male and female patients. We listened to Carol a lot for the last years, and female patients will be in the study. We want to target 3 to 20 years old patients with a distribution of the youngest patient, 30%, between 7 and 12 years old, around 40 patients, and more than 12 years old, 30 patients. These 3 to 20 years old may not be the final protocol. We may have some younger patients in the study and may have some older patients in the study. Again, it's subject to our discussion with Aurora and our group and also with the EMA. We will include patients with families present and willing to participate in the study. That's normal. Normal blood test, and also normal or near normal EKG to not have any problem with the heart. We will exclude patients that have chronic allergy rhinitis, uncontrolled chronic allergy rhinitis, or recurrent spontaneous epistaxis, since it is a nose-to-brain drug. The patient will receive nasal spray every day, so we cannot have that kind of patient. We'll also exclude patients having other diseases. Patients who receive anti-epileptic drugs will be included if they are on stable regimen. And we don't want a patient that will change anti-epileptic drugs at the beginning of the study. May happen during the study, but not at the beginning. Our primary endpoint would be a composite score based on the cognition that will assess thanks to the tablet-based cognitive and simple recording task of Aurore, and she presented it this morning. We'll also assess the behavioral disorder and emotional dysregulation using the ABC score. We'll assess the seizure. It will be based on a questionnaire. The caregiver burden, it's important, the quality of life of the family, and also the clinical global impression of improvement for the clinician. So it's the three components of the disease: the cognition, the autism disorder, the seizure, and the repercussion of the disease on the family life and the improvement seen by the clinician. This composite score, we plan to do it every 2 weeks, based on what explained this morning Aurore. With the patient should not go to the hospital every 2 weeks. However, there will be course and there will be tests on the tablets. Secondary endpoint, we will have also some efficacy endpoint. We will assess the autism using some other score. We will also fix with the family a goal attainment scale, meaning that, you know, and Aurore was talking about it this morning, can the child dress himself, and does he perform better after 6 months of treatment or not? And this is the kind of goal that can be defined with the family and can be assessed. Maybe not on every 2 weeks, but at least beginning and end of the study, that's important. And we'll use also some other questionnaire regarding the quality of life. Lighter scale, Vineland scale, we will not repeat it every 2 weeks. However, we think that at T0 and end of the study, it should be done. It has been said that it will be a great way to assess the hopefully transformation of your kid, but so we will include it in the study. Safety and tolerability, it's essential also. There will be questionnaire and nasal examination at each hospital visit. And for exploratory endpoints, Alois Mabonzo, who is not here, had a talk last September in Paris about some new blood biomarkers that we have, carbonic anhydrase 2 and PRDX6. And this is the biomarker that we test in the blood. It will be exploratory because today we don't know if they will change with the treatment. And MRN with MRS will be also exploratory because, as was saying Emil Kakis yesterday, it's probably not an enough sensitive way to see the creatine variation in the brain for this kind of treatment. However, it needs to be done because if we can see an increase in the creatine peak, in addition to a change in the symptomatology, it will be great for everybody. So this is a workflow. I have it here. Okay. Sorry about that. So you see, we want to use this placebo period in 3 groups, different, 8 weeks for the first group, 12 weeks in the second, 16 weeks in the last group. And then it will be followed by treatment during 24 weeks with this red bar at each time. The idea here is to compare 24 weeks of treatment against 8 weeks of placebo, against 12 weeks of placebo, against 16 weeks of placebo for each patient. It's not group comparison, but it's more, the idea is more to introduce randomly the treatment and see if there is a truly a change for each patient between himself, against himself. There will be hospital visit. There will be the visio every 2 weeks or almost every 2 weeks, the primary endpoint, secondary endpoint, etc., etc. This design has been used by some companies for ultra-rare disease. So we know that FDA and EMA agree with this kind of design. We will have this scientific advice with EMA after the summer to be sure that they agree with that. Where will we conduct the study? It's a question that I had during the symposium. Today, we are looking for funds. And truly, as Emil Kakis was saying, as Jerry was saying this morning, everything depends on money. And today, we plan to do it in Paris and Lyon at our hospital. It's, for us, both the quickest as well as the cheapest way. We're still talking about a multi-million-dollar study. So it's not so cheap. With more money, we may open some center in the US, but so far we don't have even it for the French study. So still raising. I would like to thank my team, which amazing and work a lot to develop this drug, and hopefully we'll be able to launch this study in 2027. And if it's positive, make sure that it accesses the market for your children. I thank you, everybody. I don't know if there's a question.
Hi. Sorry, I have several questions. I'm a clinical operations program lead in rare disease, and so I feel like I'm seeing a lot of things I'd like to have some clarity on. Thank you for answering the question about if this is a global trial. That helps. Would you be open to cross-border enrollment?
Well, it was a question that we had with Aurore. In clinical trial, we, as a sponsor, we will rely on the doctors to enroll the patient in the study, first of all. But usually what is done in France is that the patient needs to benefit from the National Social Security. The National Social Security will not pay for the study, obviously, it's not its role. However, if anything happens, the patient has to be under the National Social Security benefits.
Gotcha. Thank you. For your inclusion-exclusion criteria, you had said that the patient would need to have a normal or near normal EKG. Does this exclude patients with prolonged QT if they are stable, if their QT level has not worsened or changed?
A long QT is a problem for the heart. What has been published by Levin, we and our group did not fully agree with what they said was a long QT. So there is some misinterpretation there. And again, as a sponsor, we just need to rely on what sees the doctors. And as a cardiac surgeon, reading the Levin publication and then discussing with the French group that also, I think, published an article on it, there are some things that can be discussed, but it's, safety of the patient is key. So a very long QT will not be included.
Sure. So the answer is not no, it's maybe.
Well, as a sponsor, I rely on the doctor.
Okay.
I'm a doctor as well, but in that case, I'm the sponsor.
Okay, that's fair. And then let's see, because you have EKG monitoring in your schedule of assessments, so you can track that, right, in case it does change.
Right, we can track the EEG. However, what happened in EEG for healthy volunteer is not a demonstration of efficacy. It's a demonstration of the drug going to neurons and having an activity within neurons of a healthy volunteer. What kind of change we may see in patients will be exploratory. I didn't put the FNIRS from Laura Baroncelli, but we were discussing yesterday with her, and this is something that we can include also as an exploratory way of looking for the efficacy of the drug.
Thank you. I'll hand over.
Hi, thank you. I'm just wondering what the dose is for your phase 1, if it was a dose range finding, and what dose you're bringing into phase 2, and if you also looked at varying dosing regimens.
Well, our formulation is a 30 milligram per milliliter, and we cannot change it because the dosing within the formulation participates in the stability of the prodrug. One of the key problems of prodrug is their instability. If they are too unstable, you cannot formulate it, you cannot have a drug. If they are too stable, they don't deliver the drug. So today we are using this formulation, and we have made some formulation with lower dosing and higher dosing. And again, it was, there was a problem of stability. And based on that, with our dosing, with our spray system, we will give one spray of 70 microliters multiplied by this 30 microgram per milliliter for each nostril. And then we repeat it during the day. Our theory is that it's not useful to put a lot of dosing in the same nostril at the same time, because it will probably saturate the olfactory and trigeminal nerves. So it's more repetition during the day and repetition one day after the other that will create the passage of the creatine of the CBT101 and then the creatine within neurons and the accumulation of creatine within neurons.
70 microliters at 30 mg/ml?
Yes. So it's by heart, it's 2.1 milligram per spray. Yes. And if you do the math, you will say, well, it's not enough for the brain. But you will, we have a body of argument to say, it's working on mice, it's working on healthy volunteers. There is something happening. And then in monkeys, we had 50% of bioavailability within neurons. The key thing in this disease is that you need to target the neurons. It has been said yesterday, so I'm keeping repeating it. It's not the extracellular matrix which is important, it is the neurons. And you need to put enough drug in the neurons to have something happening. So we know that we put more or less enough drug in the monkeys, and then we know that something is happening in healthy volunteers. So now we need to test in patients.
Hi, thank you for your talk. Great to see it progressing, and then your plans for the next phase. And thank you for sharing the whole design of your next trial. You mentioned the single case experimental design.
Yeah.
And I know that you haven't received any scientific advice or pre-IND meet.
We actually had a previous scientific advice with EMA, and they said, yeah, it was probably the right design for ultra-rare disease.
Okay, so
we know that the
likely
design is okay.
It to actually have that? Because yesterday what we heard was we'll probably need a placebo control. So what we're hearing today is very different.
Yeah, and to emphasize what you just said, EMA said okay for single case experimental design, but at the same time, having a placebo control could be interesting. So, it was 2 years ago. 2 years ago, they were on both sides saying SCAD, perfect, placebo control, perfect as well. It's our duty now to argue with them, explain what is single case experimental design and why, when you compare someone to himself, a placebo control is not used. I mean, for your statistical consideration, you don't need it, so.
What would you do if EMA, FDA comes back with completely different feedback and you'll have to have placebo control? Or would you change the whole design?
we need to comply with the regulatory authorities. We will not launch the drug against them, so it's important to comply with what they say. Well, with the money that we have today, we'll probably concentrate on Europe. And, you know, it's an ultra-rare disease, and we know that EMA and FDA speak together. If we can achieve a great study in Europe with great results, and FDA doesn't run the drug, I mean, I don't know, Trump will not be here anymore. There might be a new president, and things may change. I don't know. But for this kind of disease, hopefully, the regulatory authorities may have the same way of thinking. We don't know.
Thank you so much, Thomas.
You're welcome.
At this time, I'd like to welcome our next speaker, Dr. Judy Miller. Dr. Miller is a clinical psychologist with 25 plus years of experience in developmental disorders. She holds appointments in psychiatry and pediatrics at the Children's Hospital of Philadelphia, Perelman School of Medicine at UPenn. She's the clinical training director at the Center for Autism Research, as well as the associate director for the LEND program at CHOP. She's been the principal investigator for the Vigilan observational study, which followed 50 males with CTD diagnosis over 4 years. Her talk today will focus on findings from that study.
Thank you. Okay. All right, we're getting close to the end, and we've learned a lot about clinical trials. I think what I'm gonna try to help us also keep track of is all of the challenges that we may face with clinical trials, while keeping the excitement about clinical trial development, because I think both can be true. So here's my disclosures, and we'll go from there. Okay, so I'm gonna start with the end, because we're at the, I don't know about you all, but I'm having conference brain. Does anybody have conference brain yet? Yes, okay. So I'm giving you my conclusions first, and then I'm gonna go through some slides and tell you kind of how I'm getting to these conclusions. And I've learned a lot in the past couple days too, so there may be some different things we can also discuss. Okay, so some challenges that we may find in clinical trial development is that not everyone who enrolls is going to be able to finish the trial. We've seen a few examples of that over the last couple days. The homogeneity of the sample may be important, and it's hard. And I'm going to give you some examples of how that can be, including age, including when you got your diagnosis and what happened before your diagnosis, and before you entered the trial, how some kids have much greater levels of impairment than others, and what we do with that in a clinical trial. Another thing to consider is that during a clinical trial, there will probably be other things happening. Now, there may be exclusions where you cannot take another medication, but it will probably be impossible to exclude other like behavioral therapies. You're still gonna be going to school. You're still probably gonna be getting speech therapy and those kinds of things. So knowing exactly what's happening is gonna be important. And there will be some medications that you would have to continue, and that may make you ineligible for a trial, like seizure meds or something like that. The measures that we're going to use are going to be highly imperfect, and you're going to hear a lot of different ideas about measures that could work, and it's a black box about, or it's not a black box, it's a mystery about which one is going to be right for the right trial. And hindsight will always be 20/20. So if a trial works, it will be that we made the right decision. If a trial doesn't work, it'll be that we made the wrong decision, maybe, but we may not know that in the beginning. We also don't yet know always what the first change might be or might be in a clinical trial. Like, can we maybe learn from GAMP what first signs of an improvement might be in CTD or not? And then we don't always know how long we would need to see a change from the onset of a new intervention. And it might differ depending on the intervention. So I'm gonna actually discuss two studies today. One is a pilot study that we did before Vigilan, and then Vigilan. So I'll discuss the pilot study first. So this was, I believe, in 2016, 2015. This was a long time ago. We knew a lot less about CTD than we do now. This was funded by Lumos Pharma, and it was a study to just figure out how would we even recruit kids and how do they even get their diagnosis. So I interviewed 20 families, families of 20 males, aged 13 months to 20 years. It was a very open-ended interview. They also filled out some questionnaires, but I asked them questions like, how did you get your diagnosis? What are your concerns now? And what could I glean about their presentation that might help us identify outcome points for a trial, but also how to enroll patients in a trial. Where might we find kids who might have CTD? Because you've seen the increase in the growth of this group over the last 10 years. In 2015, 2016, it was the entire population of CTD was much, much smaller. Okay, I'll talk about Vigilan in just a minute. So after doing the pilot study, we published the pilot study. There's the reference at the bottom. These are some of our conclusions from that might impact a clinical trial nowadays. So not all patients with CTD have early signs, which is a challenge. So in our group, before the first birthday, half of the kids had a feeding problem or a failure to thrive or some sort of weight or feeding challenge and a delayed milestone. So it was most often difficulty feeding or failure to thrive and missed crawling or wasn't walking by age 1. Another 20%, though, had only one of those, right? So they might have missed their crawling milestone, but they didn't seem to have trouble eating or vice versa. Another 5%, or only 5%, had a seizure with no other concerns. Okay, so some of the kids who were having trouble eating or meeting milestones also had seizures, but only 5% of our group were kids who seizure was the first sign. And then 25% of our sample didn't have any signs before their first birthday. That's going to have implications for recruitment and baseline level of impairment and how long a child develops before any intervention starts. The pathway to diagnosis in that pilot group was split. So it was pretty equally split between the parent was concerned right away and was asking and seeking a lot of evaluations and going and being very persistent. But just as many cases, it was the doctor who was concerned and the parent was not concerned. So we're a group of parents. We know when we were concerned, but just keep in mind, sometimes it's not obvious to the parents. And there were several cases where it was the provider who kept pushing the parent to keep going to the next appointment and find something else. And then another segment was kids with the onset of seizures. If you had seizures, that you might have other things happening at the same time, but the seizures is what prompted you to get to the neurologist, which got you to the geneticist. In this pilot group, the top concerns, these won't be a surprise, but it's nice to know they've kind of been consistent. First concern was independence, and then communication. And then when seizures were present, they were critical. And then when challenging behaviors were present, they were critical too. But they weren't present in everybody. Okay, so some of the implications for a trial would be that it's not apparent at birth, and it might take till at least 24 months to manifest. We've got a lot of examples of spotlight cases this symposium of kids getting diagnosed earlier than that, like at 18 months, but that's not going to be the case for everybody. So many kids don't get their diagnosis till 3 or 4 years old. If we had a trial, maybe that would help increase awareness and get people diagnosed earlier, but it might not. So at best, it's usually going to take about till age 24 months for enough symptoms to be manifest that that could be a clue that CTD is happening. The first signs are not the same for everyone, and sometimes they're very non-specific. So failure to thrive or difficulty feeding is not it happens a lot, and it's not something that would immediately make a doctor say, let's get genetic testing, okay? And when we conducted this study, the information about failure to thrive was still based on a landmark study done in, I think, the 80s that showed that most failure to thrive patients, when they got in the hospital, they could start eating and the hospital could help them eat. And the conclusion from that was that a lot of failure to thrive was poor parenting, which was still kind of the dogma, which is hard to imagine because since the 80s, you would think people would recognize we know a lot more about biochemical processes and genetic testing. There's a lot of reasons that you could have failure to thrive besides having a difficult time feeding your child. So, but thankfully, since then, AAP has come up with updated guidelines, and failure to thrive is now recognized as a potential early marker of something more biological. But it's very nonspecific. Missing your crawling milestone, how many parents are gonna lose sleep over that? Not very many, right? That is not something that parents are gonna be banging down the door of their pediatrician to get genetic testing because the baby's not crawling. But sometimes it's something very serious and urgent, like a seizure. So it's both the quality of the first sign and also the urgency and the severity of it are really different. And furthermore, you might have no concerns in the early beginning of your child's life, or you might have something really serious like seizures. So that pre-treatment issue and the timeline is going to be really variable, and that matters. The kid who's having a seizure early on might end up having a different developmental course and different response to treatment than a child who had no signs till they were 2 or 3 years old and then were developmentally delayed but never had seizures, right? Those might be two different kind of kids and presentations. So the implications for all of this are recruitment, how to find families that might be eligible, how to understand the sequelae from that pre-treatment period, how long has a child been developing without intervention before they enter a trial, and what does that do to the potential for improvement moving forward? What's the impact of different co-occurring conditions across different kids, and what's the impact of different baseline levels of impairment and how that impacts your outcome measure? We've heard a lot about cognitive testing and developmental testing, and sometimes, the standard developmental tests that we give to kids in school, like the Wechsler scales that Dr. Curie rightfully points out don't work for a lot of our kids. More and more kids with CTD are getting identified with milder presentations, and we may enter a phase where there are some kids for that test works. And comparing that kind of kid to someone who cannot do any kind of psychoed testing like that, there's a little bit apples and oranges. And it has implications for what you pick as your outcome variable. Okay, the second study I want to talk about is the Vigilan observational study. That was funded by both Lumos and then Ultragenyx. And that was 50 males. The age range was 18 months to 24 years. And that was a longitudinal study. We had in-person testing every 6 months for 4 years with lots of checks about every 2 months. I think parents would get forms. So if you think the ACD registry form is a lot, doing the same forms every 2 to 4 to 6 months, it was a lot. The families really were very generous with their time, and it was really a lot to ask, and so we're grateful for all their effort. That study was a network study, so part of the reason that study happened was for multiple purposes. One was to understand the natural history, but it was also timed to, there was an intervention being developed, and the idea and goal was that the intervention would be ready in a few years, and during the interim, we would have this natural history study that would have helped us understand the course without treatment, would have helped us understand the course in this rare disease that we didn't really know much about. It would also have built the infrastructure that you need for a trial. So part of the project was to identify other sites, identify other recruitment sources. The ACD, was ACD around in 20? I don't know when ACD started. I think it was around, but it did not have the robust group that you have now. And when Lumos approached me about doing this project, they were getting most of their we got most of our recruits, I think, from geneticists that they knew at different institutions who were seeing these patients. So it was a different world. We built up 11 sites. We had families travel from Europe and across the US to whatever site was closest to them. We changed things as we went. So we were also trying to learn what might be a good potential outcome measure or set of outcome measures. I think we did 6 protocol changes over the course of the 4 years. We tried a lot of things, and a lot of them didn't work. And we had to demonstrate that. So you've heard, we all know the outcome measure is a challenge. At the point when we were designing Vigilan and throughout Vigilan, part of the task was to demonstrate that some of the measures that you would think we should use do not work. So we were designing a study to show that it would fail, if that makes sense. Okay. We got a lot of data from that study. But if you notice, not everyone was able to finish it. Now, you'll see people just peeled off a little bit. I think we had great retention. Families really worked hard to participate. Families who knew they wouldn't be able to participate didn't join up, right? So there's a selection bias. It's only the families who really think they can do this extensive study that even gave it a shot. And they were largely successful, but not everyone finishes. Life gets in the way, things get hard, things happen. We had a pandemic in the middle, for example. But we did end up with lots of data. But it's important to keep in mind that not everyone's going to finish. We also know, as I mentioned, that therapy is happening during our observational study. Therapy will be happening during a clinical trial. So this is our data on the Vigilan group of what was happening in the meantime. So a lot of kids were taking seizure meds. A lot of kids had GI issues, behavior concerns. They were taking relevant supplements. I've heard so many more examples of kids having a positive response to supplements than I have heard for the past 10 years. So that's really encouraging to see how many kids are having that variant where the supplement can be helpful. There's allergies, there's other regular medical conditions that happen. There's a few kids with complex conditions. So things are happening medication-wise. In therapy, most of the kids were getting every kind of behavioral therapy you could imagine. So they're getting OT, PT, speech, and behavioral therapy, some combination. Most were getting all 4, and that will probably still be happening. But that's going to be different for every kid, the quality of the therapy, what the goals are. So if you think about, if you have 2 kids and they're both getting speech therapy, or they're both in a special education classroom, and they both enter a trial, you might end up with one kid, so they're on the same med, maybe they start out, I don't know what their baseline presentation is, but maybe one of them is in a very high quality program that's getting great therapy, they have a great connection with their therapist, the parents are able to implement the things that the therapist is working on, there's great communication, and you're just humming along doing really good things. Maybe there's another situation where the classroom is not as strong, or the therapist is not as talented, or the family's not able to implement the strategies as much, or it's just not clicking as easily. If you have those 2 kids in a trial, this kid might do better on the trial because maybe the medicine is making it easier for them to learn, and they're having this great opportunity to learn, where this child might not have the opportunity to learn as easily, and that might influence how they do over the course of the clinical trial. Okay, some of you have seen this slide before. These are some of the most notable milestones and events in CTD. So this has come up in the guise of what are some output points that might impact most kids with CTD, and it's probably going to be something like daily living skills like toileting and expressive communication, because so few kids make it to independent toileting and full fluent language. This is to indicate how when we started the Vigilan, one of the main measures used in developmental disability research was the aberrant behavior checklist, because it had been FDA approved. And it was a time in the world where it's still sort of that time, but it's a little bit better, where you would have to demonstrate that you would have to demonstrate why the ABC was not sufficient for your trial. So we had to use it in Vigilan in order to see, is it usable in this population? And if not, why not? so we could make the argument that it was or was not a good measure moving forward. We didn't think it would be a good measure because not every child has challenging behaviors. Many do, but you wouldn't want that to be your primary outcome if everybody doesn't struggle with it. So we used a lot of measures, and like I mentioned, we had several protocol changes. We have put together a table about what we tried, what we think worked, what seemed promising. I think it's really interesting to compare what we've done to what Dr. Curie's done in her group, because they're two different samples and different measures, often trying to capture the same kind of underlying domain, but two groups doing good work can come up with different ideas. And the core outcome set that ACD put together is really critical to kind of align people. But the next step after a core outcome set is a core outcome measure where you can really have something that is consistent. And we're not there yet. But as you've seen from the presentation just right before me, like there are people who are ready to start a trial. You have to use what's available. So you can't always just wait for what's perfect. You have to go with what you have. And the decisions that you have to make are very complicated. And it's easy to look and see what's wrong with the design. And so you have to keep in mind there's a lot of factors that you're trying to balance in order to make the best decisions you can. We tried several parent questionnaires. This was sort of fresh. I don't know what the right word is. This was very bittersweet. I knew at the outset that these measures were going to be frustrating for parents, but we had to demonstrate that they wouldn't work in order for us to do something different. So we had to hand questionnaires to parents, like the ADHD rating scale, knowing like these items just do not apply, and we know you're going to get frustrated, and we sort of need you to get frustrated so we can demonstrate that this is frustrating and it doesn't work. But we did that with several parent questionnaires. We also tried clinician rating scales. The CGI, we haven't talked too much about that, I think, in the symposium, but it's a global rating scale on a scale of 1 to 7. How is this child in terms of impairment? And then when you have an intervention, how are they in terms of improvement? It's one of the most robust kind of clinical trial measures because it's so global that it's relatively easy to show change, and it's relatively easy to get people to understand what you're trying to measure. In developmental disabilities, though, it's really challenging because am I measuring the challenging behavior that was really hard in the beginning? Am I measuring their IQ and where they may or may not have changed over time? Am I measuring their adaptive skills? Like, what is it that we're exactly measuring? So some groups have tried to come up with a CGI that's specific to their developmental disorder, and that may be a good way to go. We tried to get all of the 11 sites on the same page about how to do the CGI, and it was tricky because we were a bunch of psychologists who come with their own kind of history of how they would use an instrument like that. Am I comparing this kid to kids with no developmental concerns, or am I comparing this kid to all the kids that I know with developmental concerns? And even decisions like that are important, and you have to get everybody on the same page about them. We also used a lot of devices and wearables. We did not have success with wearables. Our kids took them off. We had caps for measuring EEG. They did not like that. We tried several things, and wearables were hard for us. Our kids were younger, and it did get better over time. So one of the things that was surprising about the trial was 6 months later, kids would come in, and you could tell they remembered us. They remembered where they were. They would light up when they saw their favorite RA. It was very cute. They would be able to do things that they weren't able to do 6 months before, and they remembered it, and they were like, "Oh yeah, I can do that now." So there was definite improvement over time, and some kids were not able to use the EEG cap or put things on in the beginning, but then were able to over time, which is great. But if you need them to come out of the gate with that cap on, you'd have to do something different to make sure that they were able to do that. Okay, well, we didn't try criterion referenced measures, which is measuring a fixed set of skills, not the general population. So for example, if you were going to use a measure like the Vineland, it would be comparing, the question might be like, are your toileting skills as good as other toileting skills in kids your same age? And you could get a score relative to other kids. a criterion reference would be like, can you do all the tasks that you need in order to use the toilet independently? Those are two different approaches, and figuring out the right way to do that is a bit of a challenge. Okay. We also didn't have the opportunity to really see if we could learn from GAMT. So could we do a pre and post study of what happens when kids with GAMT start intervention? What's the first thing to change, and how long does it take for that change to occur? Could that inform what we would do in designing a trial for CTD? Okay. So just to recap, these are some of the things that I've gleaned from these two studies about that might be challenging in a clinical trial, and we'll see what we can do from there. Okay.